This is a reply to a video response by Inmendham, or JustSayNoToKIds, link below. Some good points, I thought.
Response video to the other blubber guy. I'll leave a link below. Probably not going to be a for everybody conversation, sort of about epigenetics and evolution and genetic code and how it's read and blah blah blah. So anyway, alright, the more important aspect. I mean, first let's just agree that we're talking about a very small area for influence in terms of the evolution of an organism.
Yeah, I can agree with that.
Especially, I would argue, when I said higher mammals or more advanced or whatever I said, I think it makes a huge difference because the environment we live in is much more diverse now, and it's very hard for any effect to be effectively transferred. It would be very unadvantageous for an organism to assume something from the current conditions. From my life to the life of my prodigy could be very different.
Sure. I suppose if you had to make a guess, then you might not go too far wrong. In treating the current situation as your starting point. I agree that it's going to be an obstacle, but there are greater obstacles to overcome. As you yourself point out further in this video.
And that's sort of what's made evident, I think, is just that, depending on when the human environment is much more complex, I guess is what I'm saying, right?
Well, I guess my preference would be to sort of leave humans out of it entirely, if only because the human situation is complex for all sorts of reasons. So we reduce the sort of chances of conflating problems if we put humans to sort of one side.
Go from periods of war to periods of peace to periods of famine to periods of whatever. I mean, it's just really diverse environments that we live in. And again, we're talking about code passed on by the female, or not code, but instrumentality. And it's not very applicable in organisms that have a large amount of sexual deviation. Females traditionally lived a very different life than the males lived in many mammalian species.
Yeah, that's a good point. Though I don't know enough about epigenetics to say whether it only applies to DNA derived from the female. My understanding is that the mechanism is more of one of labelling rather than inheriting cellular machinery. So if that is the case, then there's no reason to suspect that sperm DNA couldn't be similarly labelled.
That's the truth. They're just almost entirely different lives. And so anything expressed in the female's epigenetic code might be very unhelpful to a male child. So I think there would be mechanisms put in place over time with evolution that would protect against epigenetic effects that would in any way dominate or have a controlling change in functionality. The change would have to be...
Sure. Perhaps an even greater obstacle, though, is the very delicate task of development. Selection is going to be pretty harsh on any epigenetic changes that interfere with that. Given this delicate task of development, it's surprising that this epigenetics thing works at all, in terms of inheritance.
Be or would be made to be quite subtle, I think. This damn ear is driving me nuts. Fucking can't hear. Anyway, I don't know if it's infected again or whether it's just the eardrops that are doing it, or I don't know. It's just annoying. I don't care for it. It doesn't hurt, so I assume it's not infected, but that doesn't mean too much, right? Right. Anyway. So I guess for somebody who cares to know, I hate to do instruction videos. Well, anyway, the whole idea here is that we're talking about there's a whole bunch of features of the mechanics of a cell function that's passed on from female to female. From female to everything. It's passed on by the female. That's not part of the genetic code, but it's just part of the original egg cell. And that mechanical instrumentality is, it's vital to the creation of the fetus, and but it's also just part of all your DNA is inherited. All the DNA hardware is inherited from your mother, on the female side. And, that hardware doesn't have any expression in the DNA code itself. It's not a coded feature. So it's been passed down as just hardware, almost like the membrane. You could argue that the cellular membrane hasn't been passed down. It's been augmented. It's somewhat controlled by the DNA in that features can be added to it. But the idea of it itself, the splitting of the membrane, is still a function of replication. It goes with the new cell. The new cell doesn't build the membrane. It takes a piece of the old one. So I guess that's what I'm arguing. There's some parts of you that are passed on as pieces of the original, more than just the DNA molecules. There's hardware that goes with it. And so when we talk about epigenetics, we're talking about that hardware. And a lot of that hardware has to do with reading the DNA, the chemicals that do that and interact.
Yeah, but I kind of think it's more helpful to think of it as a labeling of the code, rather than machinery used to read it. So if you've got labels that mark the genes as read me or don't read me, on or off, and then you've got sort of other labels that adjust the volume, if you like, make a gene more or less likely to be read.
With the DNA in different ways. To even prepare it for reading, you could almost argue. But I don't want to get into that kind of conversation. I'm not an expert. Look, I'm not claiming expertise on this subject, but
me neither.
I just have general knowledge, and certainly some knowledge of the human studies. That basically what it came down to was it's very hard for epigenetics to code or to create switches for any kind of positive behavior, just because our lives are so complicated. Hey, pup. Hi, pup. How you doing? Hi. Bastards. So yeah, it's very hard to code. It's very hard to express some kind of useful crude mechanism. Crude mechanisms aren't going to be very good at anticipating what we need in the future. just because our lives are so complicated. We're not bacterium. We're not living in one kind of environment where there's one kind of big change, or, you know, the patterns of our life are not the same. We have ice ages every 10,000 years. You know, we're just not interrupted by the same scale of event. And so there'd be no point in having a mechanism that didn't check against the code reader being broken or changing too many things or becoming an evolutionary feature, because it'd be much harder to fix it if it did make changes. Let's see what else helps to explain this. I mean, I brought up sperm cells before because it's kind of a funny little piece of our hardware in that they don't have any way to evolve, theoretically. The only way they can evolve is in competition with other sperm cells from another donor. You know, that's the only...
You know, I realise you're making some kind of an analogy here, but unfortunately it's sort of lost on me. But the thing about the sperm is it isn't really true. I mean, your sperm are going to your sperm are not identical. You know, they're gonna share on average only 50% of their genes with one another. So while they might gang up against rival sperm, selection is still going to favour competition between them.
Measure of a competition that could be useful because they're all coded the same. So even though they're competing with each other in a very vital competition, there's no representation of that competition in the sperm cells themselves, and therefore no way for it to have an evolutionary impact. The winning sperm cell doesn't get to express its victory in any way in terms of creating better sperm cells. Like I said, unless the competition is with the forces opposing the sperm, or, you know, from the woman, or you know, competing males, donation. So the only mechanisms they have are mechanisms that make them capable of playing some war games with competing sperm. But that's the only way they can express any evolution in terms of mutations that might take place to them. Their mutations are kind of lost in the genetic code substantially. There's no way to re-represent them. Yeah.
Yeah. Again, you know, it's probably my fault. But I still don't get what you're getting at. I mean, sperm do evolve like anything else. The genome of a sperm, you know, has to be good at being a sperm, or part of a functioning sperm, you know, before it can be good at being part of a functional embryo or a child or whatever.
Ah, okay, I'm back. You know, didn't empty the memory. Anyway, these batteries are now iffy, so who knows if I finish this or not. Anyway, I don't want to drag it out, but it's sort of an interesting subject, the whole DNA molecule and how it functions. So anyway, back to this epigenetic thing. All right, this is hardware that's, it evolves, but it doesn't evolve much. It's not carried on the blueprint. It's not part of the DNA per se. But the DNA molecule is evolving to the hardware. So that's part of the connection here that I'm getting to, is that in complex mammals, what happens epigenetically is that when the epigenetics breaks down, let's say, the more advanced features are perhaps more fragile, or there's just more fragile features that are more fragile in terms of their function. You know, compounds that exist or don't exist. You know, it's a hardware toolkit, and tools can fall out of the bucket, so to speak, of your epigenetics. And if you lose those tools, there will be parts of the DNA that have evolved, assuming their existence.
Yeah, absolutely. Yeah, that's a very good point.
And without their existence, you end up not reading code, new code. And so you don't jump to the don't grow a tail program in development, and instead you end up growing a tail. And that's what I meant by errors. That most of the effects on epigenetics, I think, in advanced mammals are just going to be ones that are a reflection of damage. They're not going to be practically useful in terms of detecting DDT in the environment and reacting to it in some way.
Yeah, though I guess, taking your previous point further, we might assume that selection will have done its work on how these labels are applied. How they're applied to the various genes. Some more important than others, maybe, or that selection establishes through the filter of selection which genes it definitely needs to reset, in a configuration that's compatible with embryology.
Again, you have to understand it. The epigenetics is a very slow evolving element of your DNA. It doesn't have this.
Well, I wouldn't have thought in terms of creating a phenotype that's adapted to its situation. That epigenetics is particularly slow mechanism. It's not going to be as fast as neurology or hormonal regulation, but it's going to be somewhere in between those and the rather slow plodding Darwinian process.
Same. It has some mitochondrial DNA. It has some DNA molecules in it that provide some of the functionality of the basic hardware kit that goes with the female to accomplish the task of reading the code. But that's really, I guess, essentially all.
Yeah, I get the the cytoplasmic kind of asymmetry thing. That the sperm contributes, a negligible. The sperm's contribution is negligible in that respect. But I really don't know. I really don't know how significant the asymmetry in the cytoplasmic contribution of the egg and sperm are going to be in this mechanism. But I guess if the labelling analogy is correct, then it should only be a very small part of it. Confined to things like the mitochondria that really have very, very few genes in them.
Well, it is. It's a code reader. And the code reader can be functional or dysfunctional. And there's going to be a lot of code on the DNA molecule itself. And those genes are going to evolve to the function of the code reader. And so, again, I'd say there's a tension between the two, and the DNA molecule certainly is going to take precedent, in terms of controlling evolution. And the hardware kit, I think, is going to remain crude in most cases, extremely crude. So I don't think there's going to be much room for, much of an impact.
Yeah, I mean, I get your point, but given the complexity of, say, the embryological process, dependent as it is on, precise timing and control of gene expression, then it's kind of difficult to think of this as a crude process.
In the more complex, environmentally diverse animals, the animals that have to adapt to a changing world, but a very complex world. We don't have too many features of our physical existence that are at all consistent. What causes stress generation to generation is very different. What we're fighting for and fighting with changes. Even physically, what threatens us is so much different.
Yeah, certainly. I suppose, given the delicacy of embryology, it's surprising that any of these acquired characteristics make it through to the next generation at all. Presumably, natural selection will have done its work, and we will only be able to observe this transmission from generation, from one generation to the next. We'll only be able to observe that in characters that don't have a huge effect on embryology, or that don't consistently fail to predict the future conditions consistently.
Because we have medical interventions. That's perhaps a more interesting subject. It's just the fact that we are not, in a practical sense, doing much productive evolution, because we've in so many ways broken natural selection, at least in how it affects the DNA molecule. Now, whether modern man has also done some damage to epigenetic function, degraded it through survival of chronically broken epigenetic samplings, it's certainly a possibility. I mean, this is the catch. Let's say something dyes the membrane of the cell blue. And theoretically, if that becomes part of the epigenetic code, in the sense that it's a cell that divides into creating the female's egg, that blueness would be inherited by her daughters, and they would carry the trait of blue membrane, that stain. So the point is that there's still evolution, but it's evolution in this crude manner, where it's not really, the test is a different kind of test. How to put it? But the point is, you could certainly, you can imagine it'd be very easy to create degrades in the system, and yet still functions barely or suitably, and that all the prodigy would be stuck with that dull functionality. The only good news would be is that the DNA would still be attempting to evolve out of any failures built into the basic structure of the code that builds the new edition and the new human. Like I said, this is probably true for all complex mammals, all mammals essentially. Their lives are pretty complicated, and there is a lot of diversity in how they live based on sex. and hierarchy. So,
yeah, I mean, with regard to your last point, yes, we do have complex lives. Presumably the basic mechanisms involved in epigenetics stem from that need for constant adjustment of the phenotype. So, I don't know, while exercising, say like walking through the woods, your muscle cells are using gene products already made and stored. While they're doing that, they're also knocking out new stuff at a greater rate than when you were at rest. So the cellular machinery is detecting signals and changing the state of particular genes, or adjusting the volume, if you like, to increase messenger RNA production. And these days, pretty amazingly, these events can be measured, in the lab, in real time. So this process is is an everyday, moment by moment part of our physiology. So I guess this is one of my acute problems that I talked about in the last video. So once the stimulus is removed, you stop walking, the process is reversed. You wouldn't expect your walk in the woods to affect the state of your genes, for the rest of the day, let alone the rest of your life, or in the lives of any offspring that you might have. But it might be possible to think of scenarios where that might be useful, or where the delay in response to the change in circumstances might be extended. If a stimulus is frequent but unpredictable in occurrence, then it might pay to have the genes in walking to work mode, even in the absence of the stimulus, and that might even spill over into the next generation. But there will be costs. So like for instance, some insecticide-resistant insects, can devote up to sort of 7% of their dry weight in the enzymes responsible for the protection that they get from the insecticides. But in the absence of the insecticides, those individuals are at a significant disadvantage to those, be it they're genetically identical, but genetically identical to them, but have no recent history of exposure to the insecticides, and so only produce trace amounts of these expensive products. So in competition with them, they're going to be at a distinct advantage because they're investing, wasting resources on these enzymes.
Some traits actually might be epigenetically controlled. I'm not sure about the changes that take place in male orangutans, alpha males. That might be something handed down by their mother based on her status in a troop. But I don't think it is. I think that might be true of bonobos. I don't think it's true of orangutans. So anyway, yeah, it has an influence, but it's not really an evolutionary influence, because epigenetic code doesn't evolve at nearly the rate at which the chromosomes can evolve. And in more complex creatures, any changes, the blue membrane isn't likely to be a major change. You're not going to get very complex changes.
Yeah, I get, but I guess like neurology or hormonal control. Like neurology and hormonal control, it is ultimately under genetic control. The genes are not influencing the moment-by-moment events, but they do determine the range of events that are possible.
They're going to be simple changes, probably of little consequence, rather than major changes of significant consequence. And because we aren't animals just swimming in a sea of 10 chemicals, you're not going to have epigenetic effects that can control for subtle chemical changes in the environment. So I just don't think epigenetics is a very interesting subject in higher mammals. It's a 0.0001% of the evolutionary interesting elements. It's a very small percentage of evolutionary significance or influence.
Yeah, I mean, I agree that its influence is limited, in that the changes don't get themselves into the permanent record like DNA does. But I imagine that, or you can speculate that it will influence the landscape in the selective landscape, and what DNA gets into the future will be part of the selective landscape. I mean, and you could speculate that it could make all the difference, between life and death, particularly when organisms invade novel environments. That if organisms had to rely on a kind of a one-to-one relationship between genotype and phenotype, then they'll be out of luck. Maybe the epigenetics give enough breathing space to allow the Darwinian mechanism to catch up and find permanent solutions to reproduce in hitherto jury-rigged phenotypes.
So anyway, I don't want to drag this out, but I ended up doing it anyway.